Good morning to you all. Our next topic is protein purification. Actually, this topic is dealing with the purification of protein from the cells. That means we have taken a gene expressed in a cell using RDT, and those cells are cultured on a large scale. Now, these are there in the fermenter. Now, next step is to take out the cells or take out the broth, and from that we have to isolate our protein. So this is actually isolation of protein. This step is protein purification or isolation. This actually comes under a topic known as downstream processing. We can say that fermenter is an equipment where we culture our cells, large scale fermenter, or it could be a small conical flask in the laboratory where do we culture our cells. So laboratory scale and large scale, there are two types of culturing. So when we take laboratory level, the type of purification, the steps of purification is different. Whereas when you go for large scale, then the steps are little bit different because of the bulk quantity of materials we have. Before going to the purification proteins, they have introduced some concepts. These concepts will help us to know better about the purification of proteins. First one is we should use only GRAS organisms. GRAS organism is generally regarded as safe. Generally regarded as safe means they are non-toxic, they are non-virulent, and sometimes it is said that these organisms are not producing any antibiotics. Next, molecular farming. In your syllabus there are two types of molecular farming. One is molecular PHA farming, and the other side it is given in the other chapter, it is given molecular farming. So here in this chapter we are dealing with the molecular farming. Pharmaceuticals. Molecular farming is production of pharmaceutically important molecules using animals as bioreactors. That means we are producing a pharmaceutically important molecule in an animal, and that animal is used as a reactor. A very innovative concept is given in your textbook, that is production of insulin in milk. Insulin are produced in pancreas. Now this gene, the gene which is responsible for insulin, is through recombinant DNA technology expressed in the udder cells of cow or any milk producing organism, and thereby we can get insulin produced in the milk. So far हम क्या करते थे, इन इंसुलिन को एक बैक्टीरिया के अंदर ट्रांसफ़र किया थ्रू RDT. बैक्टीरिया को हमने बड़े लेवल पे फर्मेंटर में ग्रो किया, एंड इस फर्मेंटर से डाउनस्ट्रीम प्रोसेसिंग करके इंसुलिन निकाल. वेरी फास्ट टेक्निक. बट उससे भी आगे हमने मॉलिकुलर फार्मिंग के कंसेप्ट लाया, वेयर नाउ द इंसुलिन इज एक्सेसिंग थ्रू मिल्क. अब ये जो प्रोसेस है इसमें क्या एडवांटेज है, व्हाट आर द एडवांटेजेस ऑफ प्रोड्यूसिंग इंसुलिन इन द मिल्क. फर्स्ट एडवांटेज है high production capacity. Generally if you produce in the case of bacteria and all, A bacterial cell produces a particular amount of insulin, and once you get that insulin, or if you have to harvest that insulin, you have to discard, or you have to kill this bacteria, or you have to go for purification process. If it is produced in other cells or milk producing cells, then their production will be very huge, because every day cow produces milk, and that too the production of milk is a continuous process, so the production capacity is very high. Now milking of cows are the process by which you have to access, or we are collecting the source material. Whereas in the case of bacterial culture, we will have to go for the downstream processing of this, which is not generally very expensive process. The third advantage is moderate capital instrument requirements and low operational cost. For growing bacteria cells, we require fermenter and all associated equipments, whereas here moderate instruments are required. That means you are actually growing cows or rearing cow or sheep or whatever cattle, and the operational cost is also comparatively low. Fermenter operation cost is very high. And the next is ease of production, including purification and scale up. For the production of this, inside the cow, the cells will have a very regulated environment. Only you have to take care of animal from the disease and all. Whereas a fermenter is to be taken care of very well, because all the internal environment is to be monitored thoroughly, and scale up is also very easy compared to the bacteria cells. So these are the 4 points for advantages of producing insulin in milk. It's a very innovative idea. We'll see what are the factors which we need to keep in our mind before charting out any purification schemes. First one is, you should know the exact source of material. That means whether your protein is present inside the cell or the protein is secreted outside. Certain proteins are intracellular, but some others are secreted outside where they are extracellular. So we should know exactly where our protein resides. That is first factor to be taken care of before designing any purification schemes. The second one is, you should know the quantity of protein required. You should know about the quantity. If you know the quantity of protein produced, then accordingly you can supply the substrates. Means if you are growing bacterial cells inside the fermenter, अगर आपको 1 kg प्रोड्यूस करना है प्रोटीन, तो कितने सेल्स चाहिए इधर, और उतने ही सेल्स को ग्रो करने के लिए कितना वॉल्यूम होगा, और कितना सबस्ट्रेट वो कितने दिन में यूटिलाइज करेंगे. ऑल दीज थिंग्स वी शुड नो बिफोर प्रोसीडिंग टू द प्यूरिफिकेशन scheme. And the third important feature that you should know is physical and chemical properties of your target protein. Then only you can design the exact techniques by which you can separate or isolate those proteins. Suppose the proteins are hydrophobic, you can develop some of the technique, you can utilize some of the separation techniques which exploit hydrophobic nature of that protein. So these are the 3 important factors that you should keep in your mind before designing any protein purification schemes. Next important thing given in your textbook is table, which is showing different steps used for extracting or purifying a protein. You can say here 1, 2, 3, 4, 5 steps. Just keep this in mind. There are 5 steps, and when you do different steps for protein purification, how do you monitor that the protein is getting purified or not? So this table will help you to understand how we are monitoring the purification of proteins. Crude extract is the starting material which we take. For example, just after fermentation you got a broth, and that broth is your crude extract. Suppose from this you are going to isolate the protein, so this is crude extract. Crude extract के total protein and that protein's activity. Come to the specific activity is total activity divided by total protein. So here you can see this many units of activity and this many mg of proteins. So 200. This is how. Suppose if you are not given specific activity, you can calculate it, provided that if you are given total protein and total activity. Now when you move from top to bottom of this particular chart, you can find that the specific activity gets increased. 600,000, 4,000, 15,000, 15,000. So just see the specific activity. If specific activity is getting increased, and that says that the purity or the protein is getting purified. So we can directly say that specific activity will show you the purity of your protein. Now you can say 15,000 ही रह गया. एफिनिटी क्रोमेटोग्राफी एक टेक्निक है, साइज एक्सक्लूशन क्रोमेटोग्राफी दूसरा टेक्निक है. ये करने के बाद 15,000 था, साइज एक्सक्लूशन करने के बाद 15,000 ही रह गया. दैट मीन्स इसमें एक्टिविटी कुछ बढ़ा नहीं. तो वी कैन से दैट ये साइज एक्सक्लूशन करने से हमें ज़्यादा कोई प्यूरिफिकेशन नहीं मिला. वेयर एज 4,000 से 15,000 हो गया एफिनिटी क्रोमेटोग्राफी करने के बाद. 4,000 became 15,000. That means affinity chromatography is a very effective step. आपके एग्ज़ाम में 2 क्वेश्चन आता है, विच इज़ मोस्ट इफेक्टिव. मोस्ट इफेक्टिव स्टेप कौन सा है और टेक्निक कौन सा है, और लीस्ट इफेक्टिव स्टेप कौन सा है, ये पूछेंगे. तो मोस्ट इफेक्टिव स्टेप समझने के लिए सबसे ज़्यादा स्पेसिफिक एक्टिविटी कहाँ पे हुआ है, दैट विल बी द मोस्ट इफेक्टिव. हियर एफिनिटी क्रोमेटोग्राफी. क्यों साइज एक्सक्लूशन नहीं है? क्योंकि एफिनिटी क्रोमेटोग्राफी में 15,000 था, साइज एक्सक्लूशन में उतने ही प्यूरिफिकेशन, सॉरी उतनी ही एक्टिविटी रह गया. मतलब देयर इज़ नो फर्दर प्यूरिफिकेशन हैज़ बीन डन. सो लीस्ट इफेक्टिव विल बी साइज एक्सक्लूशन. क्लियर. एंड वन मोर क्वेश्चन कम्स, इस पर्टिकुलर प्रोसेस की यील्ड कितना है? यील्ड कितना है? व्हाट इज़ द आउटपुट? आउटपुट कुछ नहीं, यू कैन डायरेक्टली गो टू द लास्ट स्टेप. 45 mg इज द यील्ड ऑफ दिस प्यूरिफिकेशन स्कीम. ये भी आपके टेक्स्ट बुक में दिया है, जस्ट गो थ्रू इट, विल हैव सम क्वेश्चंस लेटर ऑन. अगर ये 15,000 कहीं पे भी इक्वल वैल्यूज़ नहीं, सेम वैल्यूज़ नहीं है, देन यू कैन से दैट सबसे कम हुआ है प्रेसिपिटेशन बाय सॉल्ट. ओके, बस सबसे कम स्पेसिफिक एक्टिविटी वहीं पे. अगर क्रूड एक्सट्रैक्ट को क्यों नहीं यूज़ करते? क्योंकि क्रूड एक्सट्रैक्ट तो पहला ही हमें मटेरियल है जिससे हम स्टार्ट करते हैं. तो क्रूड एक्सट्रैक्ट इज़ नॉट ए टेक्निक. सो यहाँ से टेक्निक स्टार्ट होता है. क्लियर. ओके, we'll move on to the next. Absorbance at 280 nanometer is one of the easy, fast, non-destructive technique for assessing concentration of the protein.